Hi everyone, welcome again to the course of structural biology. We are continuing with the module of cryo-electron microscopy. Today, in the previous 3 classes, I talked about microscopy, I talked about cryo-electron microscopy, different aspects, their sample preparation, their instrument details, the plunging, the high performance freezing and all these things, data collection, data analysis. Today, I want to summarize that in a form of a story. I hope you will like it, because cryo-electron microscopy among the 3 high resolution techniques is considered as the most potential one. How the technique have traveled, what are the challenges, what are the journeys, I am summarizing them in a way you could see how it comes up. So, a concise story of advancement of cryo-electron microscopy. We will start with the beginning days, and there is a talk of a science fiction which happened in 1968. George Gamow, a physicist, and Martinus Icas, a microbiologist, they published a popular science book called Mr. Tompkins Inside Himself, The Adventures in the New Biology. This is the book. What this book tells about? This book tells a story about Mr. Tompkins as this guy explores the cellular architecture of his body on a dream journey through his bloodstream, guided by his doctor. It is a very amazing story to learn about our body and system, how it works. But the more interesting thing and more context to today's story is, while inspecting the structural details of single cells and organelles, Mr. Tompkins' guide enthusiastically informs him that this knowledge is based on studies using the electron microscope. Mr. Tompkins' tour in the body makes it obvious that at that time the instrument have already brought studies of biological material to be a previously unimaginable level of detail. So, already people have started understanding the importance of electron microscopy. However, until just a few years ago, scientists could still only dream of being able to use the electron microscope to zoom in further into cells and organelles in order to uncover the atomic details of the biomolecules that underpin their architecture and the function. So, yes, overall biology, it was possible, but entry to the atomic level was kind of impossible, challenging. This dream became reality recently when a series of critical developments made it possible to take full advantage of the pioneering discoveries and improvements made by the Nobel laureates Jacques Dubochet, Joachim Frank, and Richard Henderson. We talked about them, we will talk about them in further details while going through the stories. These advances now allow structural determination of non-crystalline biomolecules in solution at high resolution using single particle cryo-electron microscopy. Through this journey, I have repeatedly talked about how important it is to get the 3-dimensional structure of the biological macromolecule, especially protein. But the challenges we have already discussed about crystallography. It is a very convenient technique. It have gone through long way, but it still needs the basic thing, basic requirement is to get a crystal, and the possibility of getting crystal for a protein is really less. Now, with the further advancement of the technique of cryo-electron microscopy, you get the opportunity where you use near native condition to get the structure. Obviously, with your new knowledge of NMR, you will question me about what is happening in the NMR. Yes, as I always agree, NMR is another very potential instrument which we have to explore much more than what it is there. But in the current state of art, it is really difficult to get the protein in that high concentration that NMR demands. Moreover, to get the structure, you have to go to the 3-dimensional, 4-dimensional experiments, and it is challenging to get that because it is expensive. You have to get only the hydrogen isotope which is NMR sensitive, NMR active is natural, others are all you have to provide. So, that makes the cost of the growth of the protein in a huge high, and addition to that, in the presence of this unnatural isotope, in some cases protein also do not want to grow. So, this is coming. Continue with the study sought after the experimental demonstration of an electron microscope by Ernst Roorkee, which we talked about, for which he was honored with Nobel Prize for Physics in 1986. Ladislas Martin published a paper that commented on Roorkee's discovery. Why that paper is important? In this short report, Martin noted that the new instrument, which is the electron microscope, unfortunately could not be used to study the biological material without the destruction of the organic cell by the intense electronic bombardment. So, what he is trying to convey the message is, with the electronic bombardment, which is the integral part of the instrumentation working, it is not possible to make the biological materials intact. It will destroy the material. Preventing such destruction would require a new sample preparation technique. He is the first one, Martin is the first one who first talk about that with alternative sample preparation we could have start getting information about the biological sample using electron microscope. Martin proposed visionary solution to this great problem. Many things that it is the starting milestone of today's cryo-electron microscopy, solving atomic structure. So, strategy, the solution, cooling the biological material or the use of an approach similar to negative staining. So, he was first talking about cooling, the key concept to cryo-electron. Another major problem was how to preserve water in the biological sample in the vacuum maintained inside the electron microscope chamber. So, we always want the protein in solution. So, how to preserve the water? This is extremely important in the biological sample in the vacuum condition maintained inside the electron microscope chamber. And there are even other challenges to face. To mention only to the most basic ones, intact biological material has very low image contrast. Why? Because most high energy electrons pass straight through the specimen would not make any effect. So, it have low image contrast. At the same time, the electron dose must be kept low enough to prevent damage. So, one, preservation of water is a challenge. A solution was cooling, and then biological sample have low image contrast, and electron dose is important because higher dose damage. Now, if you make lower dose, contrast or signal by noise is even worse. The probability for multiple electron scanning events must be negligible. If you see, we talked about when electron hit a material like light hit material, a lot of effects. So, multiple electron scattering events must be negligible at the electron energy used sample. So, to do that you have to have thin samples, ideally single layer. Furthermore, the studied objects often move both upon interacting with electrons, and due to drifts in temperature, the movement reduces information content, especially when using film or slow detectors to record images. This we have discussed in detail in the last portion. So, I am not going into details, you already understood that in time of data collection. As a result, the resolution was typically limited to a few nanometers for biological molecules, while atomic level resolution was required to see the functional biological macromolecules. So, we need it in angstroms, and this is what correct, we get it in nanometer, not correct. So, you see the earlier ones, but then it start improving and we are going literally to the higher resolution, as you see here the atomic details. That is the journey of cryo-electron microscope. So, the negatively staining biological material or negative stain play a very important role even today towards sample preparation, checking the sample and initial development of the 2D model. The necessity to use the lowest possible electron intensities to study low contrast sample stimulated the development of new sample preparation methods when recording images of biological materials. The first commonly and successfully employed method was negative staining, established in the 1940s and refined during the next 20 years with their alteration of the material, how to use them, how to prepare them and all these things. When using this approach, the biological material is embedded in a thin amorphous film of a heavy metal salt, which generates a cast around the object. The cast scatters electron, we have already talked about this, more strongly than the encapsulated material, is more resistant to electron damage and prevent collapse of the biological material during drying in the vacuum within the electron microscope. So, the cast scattered electron more strongly than the encapsulated material. So, you have the encapsulated material and the cast. The cast is more resistant to electron damage. So, it is not directly coming to the biological sample, and it also prevent the collapse of the biological material during the drying in the vacuum within the electron microscope. So, this is, you could see the side view, you could see the top view, the electron beam is coming, the stain is protecting, right, and the thing is made on a supporting film. This is a negative staining of T4 bacteriophage. This is a negative stain of recombinant rabies virus nRNA rings. So, by doing negative staining, you start looking at the thing, you start getting better resolution, because one, it protect from damage, in addition to it scatter right more strongly, in addition protect and also protect from high vacuum setup. The approach of negative staining offer detailed information about the morphology of bacteria, virus, organelles, as we have shown just. However, for studies of single molecules or molecular complexes, in the best case the pictures could reveal only the envelope of the covered particle with a resolution that is limited by the granularity of the strain. So, we are still unable to break the barrier of atomic resolution. Nevertheless, the use of this sample preparation technique offered important low resolution structural information, as I told, for development of basic 2D material, 2D model and all, to check the homogeneity of the solution and all. This is very good. The experimental and theoretical tools used to calculation of three-dimensional structure from two-dimensional projection in the electron microscope establish the basis of today's advancement. So, through negative staining, the development of the experimental and theoretical tools which used for calculation of a three-dimensional structure from two-dimensional structure. We talk about this, we talk about how a 2D picture, different 2D pictures averaged, and then using the tiltation, using those changes, we have developed the 3D material. Aaron Klug, the Nobel awardee of 1982, noted that verification of a 3D structure of a negatively stained particle from analysis of an electron micrograph require observation from different directions. So, this concept, it require observation from different direction, is the basis of 3D development. Such an analysis could be achieved either by tilting the specimen. Tilting is a very important part in cryo-electron microscopy, or by analysis of many particle position at different orientation. So, one thing is tilting, another thing is orientation. We have talked about this earlier too. In the early 1960s, Klug and his colleagues designed optical methods for analysis of electron micrograph of periodic structure. So, from periodic structure you could have developed models. They have established method to obtain structural information from electron diffraction of thin catalase crystals enzyme in the electron microscope. Important contribution were also made around the same time by Walter Hope, who established new method in electron crystallography to analyze the protein structure. Hope also analyzed the non-crystalline macromolecules without symmetry, but the experimental approach used in these studies could only yield low resolution data, telling the importance of symmetry also. The approach could not be generated because of too high accumulated electron doses that could be potentially damaging for the sample. In 1968, David de Rozier and Klug presented the first successful calculation of a 3D material model from analysis of a 2D projections in an electron microscope. So, the first time the dream of conversion of a 2D picture to a 3D model is successful by de Rozier and Klug. The researchers have analyzed the tail of the bacteriophage T4, which was chosen because of its helical symmetry. As I told, symmetry is extremely important here. The 3D model could be calculated from an analysis of a single projection, because the information content was equivalent to that obtained from projection of 21 different orientation of each of the subunits. Calculation of 3D models of non-helical particles require combination of data from several 2D projections of these particles in the electron microscope. One approach used to determine the relative orientation of the particles that generate specific 2D projection was presented in 1970 by Anthony Crowther together with the other people which we talked about, the de Rozier and Klug. The method they used to call common line approach was used to determine a 3D envelope of icosahedral viruses based on an analysis of 2D projection in the electron microscope. The researchers suggested that the method could also be applied to particles with no symmetry by tilting the sample, but it proved to perform best for symmetrical particles such as the icosahedral virus, which maintain amazing symmetry in the structure. Calculation of 3D models of non-helical particles require combination of data from several 2D projections. So, we have the micrographs, we have the pictures of different 2D figures, and we get the projections and make the 3D model. One approach used to determine the relative orientation of particles that generate specific 2D projection was presented in 1970 by Anthony Crother together with the de Rosier and Klug, the other people who were continuously working on this. Now, coming to native protein crystal at room temperature, the techniques was also needed to preserve intact biomolecules in the hydrated state in the electron microscope and to determine condition for non-destructive irradiation. Donald Parsons developed environmental chambers in which a humid atmosphere was maintained at room temperature in the electron microscope. If you remember, we talked about different such setups now, and you get them humid or you get them auto control humidity. So, that was the first time Donald Parsons did that. Using this approach, he demonstrated electron diffraction from catalyst crystals and showed that it is possible to find conditions under which the protein structure remain intact during the electron irradiation. Robert Glaisher quantified electron induced radiation damage in studies of crystalline catalysts, and small organic molecule contributed very significantly in further advancement of the field. He realized in the early 1970s that for unstained specimens, care must be exercised to maintain low electron doses in order to minimize damage due to inelastic electron scattering. Glaisher also noted that using low electron doses requires averaging over ensembles of particles to increase the signal to noise ratio. In other words, the radiation would be distributed over multiple copies of the same particle, and that would be good. Important progress was made in the mid 1970s by Henderson and Nigel Unwin, who developed a new preparation method that enables studies of unstained protein crystal in the electron microscope at room temperature. Henderson and Unwin replaced water with a glucose solution and preserved sample in vacuum and used the new preparation method in studies of intact protein crystal. So, kind of, if you see when you are doing in crystallography, you use glucose solution as a cryopreserver. So, Henderson, who actually started with initial studies being a crystallographer, get the idea there and use that. Radiation damage in the electron microscope was analyzed systematically, and the electron intensity was adjusted at low level to minimize the effect of electron radiation. In an initial study, the authors presented projection maps of thin catalyst crystal and 2D crystals for bacteriorhodopsin in a purple membrane. So, if you do not remember, 2D crystals are crystals, protein crystals who grow flat, I talked about. So, they do not have the dimension, and when we are talking about bacteriorhodopsin, you would remember bacteriorhodopsin, the first protein Richard Henderson have come up with the structure using cryo-electron. Images of structure and of the diffraction patterns were obtained. The phases were calculated by Fourier transformation of the structure images. In a second study, the same pupil tilted the specimen as contributed by Klug to collect projection images from different directions. Using these data, they obtained a 3D map of bacteriorhodopsin, which revealed the general architecture of the protein, but still at a rather low resolution. Analysis of the electron micrographs and calculation of the 3D map from 2D projections were based on method developed by Klug and colleagues, combined with the use of information obtained from the electron diffraction pattern. So, one, they have used the tilting and all those processes which Klug have developed and added the electron diffraction pattern. The very low contrast of 1% limited the new preparation method to diffraction studies of crystal. Cooling the specimen was expected to reduce water evaporation and to protect the biological material from radiation induced damage. Now, starting in the 1950s, Humberto Fernandez Moran explored the possibilities for freezing samples and preparing thin cryo sections for studying using Cryo-EM. However, upon freezing, water typically nucleates to form crystalline ice, which strongly diffracts electrons, thereby effectively obliterating means making the process, the signal, you do not get it which is originated from the sample. So, what happened, like you have the sample and now you do the freezing. Freezing gives you ice crystal which diffract very strongly at around near to 4 angstrom. So, you get the signal coming from the water more than the signal coming from the sample. That was the problem. Furthermore, formation of ice crystal may also change the structure of the specimen. So, according to Paul Smith's historical overview, when he looked back on the development of cryo-electron microscopy, Basil Louet had already noted the problem associated with the formation of crystalline ice when cooling the cell in the 1940s. Louet realized that the solution to the problem could be to cool the biological material sufficiently rapidly to preserve water in a liquid amorphous state referred to as vitrified water. So, Louet already had isolated the problem and understood that development of vitrification of vitrified water would be a good solution. Cooling techniques were also introduced early for studies of protein crystal in the field of X-ray crystallography. So, if you see that the cooling, freezing, those techniques are very similar in protein crystallography and cryo-electron microscopy, where the formation of crystalline ice was prevented by using sucrose or glycerol as a co-protectant. So, we still use that when you are freezing, you need to freeze the crystal, right. When you are freezing, we use the cryoprotectant, because without the cryoprotectant, we will actually freeze the water, and water is a good diffractor. In the electron microscopy field, Kenneth Taylor and Glacier demonstrated electron diffraction pattern from unprotected frozen catalyst crystal to higher than 3 angstrom resolution is a revolution. They showed that hydration was maintained in the electron microscope at cryogenic temperature and contrast was improved compared to when using the glucose kind of as a cryoprotectant. The available technology at the time only allowed studied from approximately minus 120 degree centigrade, which is above the transition temperature from amorphous to crystalline. However, the researchers did not observe crystalline ice in the protein crystals, which they attributed to interaction between water molecule and the protein surface. Taylor and Glacier also developed technical solutions for specimen handling at cryogenic temperature. They showed that cooling result in improved resistance to radiation damage, such that longer exposure time or longer electron intensities are quite possible in this case. In other words, they concluded that cooling the specimen would increase the information content positively. So, this is the basic development, and now we will go where we want to go, where is Cryo-EM today, is the Cryo-EM structures at high resolution. The year 1990 marked a critical milestone when Henderson and colleagues showed for the first time that it is possible to obtain high resolution structures for biomolecules using Cryo-EM through averaging over many copies of the same object. In their experiment, the signals originated from many bacteriorhodopsin molecules in a 2D crystal. The study showed that at cryogenic temperatures, the consequences of radiation damage could be limited to retain the information content sufficiently to reveal position of amino acid chains of the protein. So, it is possible, the possibility comes in subsequent years. A similar approach was used to determine high resolution structure of, for example, light harvesting complex, the tubulin dimer, the aqua protein, which are solved in high resolution. In their pioneering study, Henderson and colleagues used several electron microscopes around the world to optimize the data quality. They identified a number of technical limitations of these microscopes as well as challenges associated with sample preparation, which at the time conspired to limit resolution. So, what they did, they have one sample and they travel around and they get access to different microscopes which are that time used, and try to make a comparison and finding out the limiting factors. Henderson concluded that no microscope was perfect at that time and argued that specific technical and specimen preparation improvements would facilitate development of cryo-EM to a general technique. This would then turn the technique we have been using into a routine and quick method able to be used to many more difficult specimens, eventually including non-crystalline molecular assemblies. So, he was kind of a visionary in this case. The first high resolution model of bacteriorhodopsin was based on analysis of millions of protein molecules in a 2D crystal, which allowed the spread of the total electron dose over a large number of particles. The analysis of a large number of molecules in the 2D crystal is equivalent to averaging directly in the microscope. For non-periodic assemblies of symmetrical particles, the signal to noise ratio can be increased by averaging over the asymmetrical unit, because the symmetries give redundancy of the signal here. However, the general case of non-periodic asymmetrical particles, the challenge was to determine the position and orientation of each particle in an image from weak signals. Once this could be achieved, averaging is quite possible. However, such analysis would require computer power well beyond which was that time available in 1990, because it's so many micrographs you have to do a simulation. 5 years after the solution of high resolution structure of bacteriorhodopsin, Henderson presented a quantitative analysis of challenges needed to overcome in order to determine atomic resolution structure of non-crystalline molecular assemblies. He concluded that by using low intensity non-destructive electron irradiation in phase contrast electron microscopy, it would be possible to determine the 2D position and 3D orientation of individual particles given a sufficiently high molecular weight. The information would allow averaging ensembles of randomly distributed particles, thereby eventually reaching atomic resolution. So, the first initiative when we are on the track. The conclusion from Henderson work was that assuming a molecular weight higher than 50 kilodalton, it should be possible to align and average a reasonable number 104. So, 50 kilodalton, his condition was higher than 50 kilodalton molecular weight, average with number of 104 of particles to determine the structure is possible at 3 angstrom. So, 50 kilodalton 104 particle 3 angstrom resolution. So, here is a comparison for large viruses where the mass is 300 megadalton. It is possible for small virus 11 megadalton possible, for ribosome 3.3 megadalton and 1.4 megadalton, for eukaryotic and prokaryotic possible, multimeric enzyme 420 kilodalton, 180 kilodalton, 52 kilodalton, yes possible, small protein or very small protein peptide, it is not. So, this data addresses the question, can single molecule alignment be carried out in practice? So, the answer is given to the right for the number of example proteins listed to the left with molecular weight in the middle column. But now you see here, I would talk about this which is called resolution revolution in cryo-electron microscopy. So, examples of structures determined using cryo-EM, the figure illustrates the size. You will see that 11,200 kilodalton, dengue viruses solved at 3.6 angstrom, P97 540 kilodalton solved at 2.3 angstrom, ribosome 2300 kilodalton solved at 2.9 angstrom, beta galactosidase 465 kilodalton solved at 2.2 angstrom, GDH 334 kilodalton 1.8 angstrom, LDH 145 kilodalton 2.8 angstrom, and IDH 93 kilodalton is solved at 3.8 angstrom. Also, now the smallest one is hemoglobin, which is solved at 64 kilodalton size. So, during the coming years, the predicted number of particles necessary to reach this resolution was revised to smaller number, and Glacier presented an analysis that suggested that the size limit could be adjusted to 20 kilodalton. If this is possible, cryo-electron microscopy will be again prospering, getting more covering area in drug designing and all these things, and analysis along the same line, but less detail were also presented in an earlier work, giving possibility of lower molecular weight protein structure determination. Coming to the ensembles of single asymmetric particles in solution, a fundamental problem in studies of unstrained non-crystalline asymmetrical randomly oriented particle in solution is the alignment of the feature that are only faintly visible on a noisy background. In the mid 1970s, Frank addressed this problem in a study that became in many ways the starting point of future developments. Joachim Frank and his colleagues presented a method of aligning low dose images of individual molecules using cross correlation function. This is again a very important part of revolution of Cryo-EM. A quantitative analysis of the problem was presented in 1977. The analysis concluded that it would be possible to look at randomly positioned particles using non-destructive electron doses. Consequently, the implication was that it would be possible to average images of many radiation sensitive particles to eventually obtain high resolution data. The feasibility of the approach was illustrated in studies of negatively stained glutamine synthase. For a non-crystalline specimen consisting of uniform particles, the challenge is to determine the position, orientation of each particle, that is the 5 parameters that determine their 2D position in the plane and their 3D orientation. So, 2D position x and y and 3D orientation. However, biological samples are rarely structurally uniform and many contain impurities, as we know. Therefore, another requirement is to identify potential structural substrates to identify different type of particles for heterogeneous samples, and the case of stained particle to identify differences in the negatively stained structure. So, if you look at them after negative staining, and you could do the classification in state initially and then on the data. In 1981, Frank and Marine Van Hill presented a method that allows sorting of particle images into classes based on their orientation as well as their structural feature. Each particle image represented by intensity of n pixels is described by an n dimensional vector. Multivariate statistical analysis is used to sort the vectors which appear in clusters. Each of these clusters is assumed to represent a 2D projection of a particle with specific orientation and structural properties. The approach could be computerized to automatically sort images based on their subtle difference in orientation. Particles within each cluster are assumed to be sufficiently similar to allow averaging to increase the signal to noise ratio. So, if you see here, it is the graphical representation of a method of particle sorting. Here each particle image, here it represented in 3 colors, 1 particle, 2 particle, 3 particle. So, represented by 3 pixels, the intensity of each pixel is translated into an integer number and the particle is illustrated by a vector. So, if you see for the red, the pixels are collected, pixels are converted into the integer and separated. Another challenge is to determine how the 2D classes are related to each other in a 3D for a given structural subset. A general method is to determine the relative 3D orientation from classes of 2D projection of asymmetrical particles was presented by Frank and Michael Radermarker in 1986-87. The method is called random conical tilt. The method is based on the general idea of obtaining 3D information from 2D projection presented earlier by Frank and colleagues, combined with the application of a tomographic conical tilt series described by Radermarker. This is the one. So, A, B, C 3 part of the picture, this is called random conical reconstruction. So, the randomly oriented particle in the A are projected in B by tilting 50 degree. Okay. The images in B which you are looking at form the conical tilt series. In C, they are represented as a single particle that is randomly projected with all directions lying on the surface of a cone. So, that is why it is called random conical reconstruction, taking from 2D, doing a tilting development of a cone which is giving you the projection from 2D to the 3D. Frank developed many of the important mathematical tools used to image analysis which form the basis of single particle cryo-electron microscopy. He gathered them together in a suite of computer program called Spider, making them readily available and usable to the scientific community. Spider is system for processing image data from electron microscopy and related fields, is an image processing system for electron microscopy. So, we have already discussed about the importance of sample preparation, how the sample preparation happened, what are the procedures and all. So, as we have also discussed in the previous section, the cooling was expected to solve many of the complications that limited the use of the electron microscopy for structural studies of biomolecules, especially protein. The problem associated with the formation of crystalline ice could in principle be overcome by cooling liquid water into a vitrified state. So, cooling is one of the solution, formation of ice is a problem. By development of vitrification, you could solve that. However, before 1980, whether bulk water could be transformed into a vitrified solid state was still controversial, because theory predicted that the required cooling rate would be practically unattainable. If you remember, when I talk about vitrification, I talk it is a huge speed. So, before 1980, people thought that this is unattainable. The phenomenon had been demonstrated, but only for condensation of water vapor at cold metal surfaces. In 1980, the discussions were brought to an end with the demonstration that vitrified water could be formed by rapid cooling of micrometer sized droplet of bulk water. It is also interesting to note that vitrified water may be the most common form of water in the universe. So, what we want to achieve is actually the most common form of water. In 1981, Dubochet and Elsdeer McDowell finally presented a method that allowed formation of a film of non-crystalline solid water on a specimen grid for observation in the electron microscope. Water was sprayed on a carbon film mounted on a grid, after which the grid was rapidly immersed in a liquid ethane or propane at about minus 190 degree centigrade, which is the temperature of the liquid nitrogen, maintained by cooling in liquid nitrogen. The thin layer of the vitrified water was shown to yield nearly uniform absorption of electrons in the cryo-EM, which was the dream. The amorphous structure of water was converted into a crystalline form upon warming to about minus 140 degree centigrade. Dubochet and colleagues noted that vitreous ice could be maintained around the specimen for extended time if the temperature was kept below minus 160. So, you do the freezing and maintain minus 160, you could maintain the condition. In the years following, Dubochet and colleagues developed the sample preparation method further and presented detailed studies of pure water, aqueous solution and suspension of bacteriophages, purple membranes and DNA at cryogenic temperatures. The full potential of Dubochet's sample preparation method was realized in 1984 when a group presented electron micrographs of virus suspensions cooled using an improved method that allowed preparation of thin unsupported water layer in the vitrified state. The new technique made it possible to prepare unsupported water layers that could be made sufficiently thin to allow rapid vitrification, but thick enough to accommodate a single layer of randomly oriented molecules or molecular complexes in their native state, which was the DIM for long time. How the sample preparation was made? The sample, you see the sample is transferred in the grid, and then the grid is coming to the liquid nitrogen cooled ethane, and then the grid is coming to be prepared under microscope. The sample in the step 1, the sample is transferred to a grid which consists of a small holes of the carbon films. You could see here, this is the grid. These are the small holes, and if you see, this is the sample where the biological macromolecules are there. So, the sample is transferred to a grid which consists small holes in the carbon film supported by a metal frame, Excess solution is removed. I have discussed it in the sample preparation section previously. When you have excess solution, you do blotting or something, you remove it. Coming to step 2, a thin film of randomly oriented particle in water spans the grid holes. The grid is then rapidly plunged into the ethane cooled by liquid nitrogen. Coming to the step 3, a film of particles in vitrified water is formed across the grid holes, and you see when you keep them under the microscope what is the picture or the micrograph coming. Dubochet and colleagues showed that the sample preparation method is generally applicable to cryo-EM studies of other biological particles as well. The resulting images showed an impressive contrast. Finally, in the beginning of 1980s, Dubochet had solved the problem that had cast a shadow over the first 50 years. Since the invention of the electron microscope, the most abundant constituent of living things, water, has invariably been excluded. So, this is the final kind of step. The new preparation method was immediately adopted by Dubochet's colleague and is now used universally within the cryo-EM field, both in studies of assemblies of single particle and in cryotomographic studies of single objects. So, that is all about the process details. Coming to the summary, the first high resolution structure determined using cryo-EM was presented in 1990, the structure of bacteriorhodopsin by Henderson. A decade passed before high resolution structure of helical and icosahedral particles were imaged, determined based on analysis of data recorded on film. After the introduction of the new direct electron detector in 2012-13, which is one of the landmark, and the first reports of de novo atomic structural model of smaller single particles such that for membrane protein TRPV1 ion channel, cryo-EM has very rapidly become a major new tool in structural biology for determining atomic structures. You could see here the structure of glutamate dehydrogenase is showing you the tour, because this is one of the stable protein which is taken many time to make the micrographs and analyzed. So, these blobby structures are resolution before 2013, which is improved now, and you could easily see the atomic level structure. So, the resolution progression of cryo-EM illustrated by a representation of glutamate dehydrogenase with an increasing level of detail in the left to right. For a protein of this size, which is 334 kilodalton, the 1.8 resolution to the right could only be achieved after 2013. It is captivating to think about the amount of time that has passed before we could get to this point. About 6 decades after John Kendrew and Max Perut's pioneering crystallographic work on myoglobin and hemoglobin, as I have discussed in details, Nobel Prize for Chemistry in 1962 for their studies of the first structure of the globular proteins, and 4 decades after the first development of the laid the groundwork for single particle cryo-EM, a high resolution structure of hemoglobin. So, hemoglobin, the first structure, now again come in solution determined using cryo-EM. Single particle cryo-EM is unique in that it does not require crystallization. As I told for a long time, this is a criteria, uses very small amount of material and covers a wide range of sizes from particle the size of hemoglobin 64 kilodalton to very large particle up to megadaltons. So, in that point you could compare NMR, crystallography and cryo-EM. Cryo-EM, it was like from more than 50 kilodalton to several megadaltons. Cryo-EM. X-ray kind of covering from low to this. If you have a crystal, NMR is kind of limited to 60 kilodalton, still now 60 kilodalton. So, where this is started, NMR ends. So, this is one of the good comparison. From small peptide to 60 kilodalton protein, NMR is good. For any peptide to good size of the protein of few 100 kilodalton, X-ray is good. From 50 kilodalton to several megadalton, cryo-EM is good, but cryo-EM works with the native condition, identifying the conformation and all in the atomic level. X-ray needs crystal, NMR needs very high concentration, none of them are required for cryo-EM. Cryo-electron tomography is used to determine structure of even larger objects, including organelles, cells, with the potential of being able to obtain high resolution information from molecules or complexes in situ. Thus, with the recent development, cryo-EM extend the possible size range of structure determination in solution from cells and organelles to molecular complexes, molecules and the atoms that build these molecules. It is a amazing range it covers. But cryo-EM is not only about static structures, because sample preparation for cryo-EM involves instant cooling of a solution. The content of the solution can be systematically varied. Integral membrane protein may be studied in a near native environment, and the particle may be trapped in structural substrate or even in action, while an enzyme catalyzes chemical reaction. A lot of realistic biological conditions could be trapped. That is the potential of the upcoming instrument. Also, one thing I have to repeat about membrane proteins. As you have already understood, soluble proteins are proteins which have hydrophilic amino acids. Hydrophilic amino acids which form hydrogen bond with the water and solubilize. For the membrane proteins, they have hydrophobic residues at the surface, so they could not be solubilized. So, to solubilize they need perfect condition, and that is why it is really very difficult to work in both crystallography and NMR not here. You could directly get the membrane, freeze it and get the structure. Membrane protein and related developments are possible with the advent of cryo-electron microscopy, like high resolution cryo-electron microscopy. The data may offer functional information, structural changes may be monitored and free energy landscape could also be determined. These recent developments will certainly be followed in time by others, both in regard to technology and applications. Perhaps in the future we will be able to obtain high resolution structural information of molecules as well as to observe interaction and dynamic process as they happen inside the cell or organelles. Probably we could get the functional mechanism of the entire cell, which would be amazing. During his journeys in 1968, we are going back now, the fictional Mr. Topskin asked his guide about the electron microscope. Is it true that it is so powerful that you can see atoms with it? The guide smiled and declared, not quite, but you can see the larger protein molecules and this is impressive enough. A question and related answer, a doubt. Very interestingly, the whole scenario was coming in the life of Professor Henderson, and we will finish our story talking about that. So, in 1997, when Henderson attended the annual Gordon Research Conference on 3D electron microscopy, a colleague opened the meeting with a provocative statement, cryo-EM was a niche method. He said, unlikely to ever supplant the X-ray crystallography. But Henderson could see a different future and he fired back a salvo in the next talk. I said, we should go for global domination of cryo-EM over all the structural methods. In the years that followed, Henderson, Agard and other cryo-EM evangelists worked methodically, as we have already described the history, on technical improvements on electron microscope, in particular on better ways to sense electrons. Long after digital camera had taken the world by storm, the researcher developed a new generation of direct electron detector that vastly outperforms both the film and digital camera detectors. So, the innovation of direct electron detectors, as I have told in the course for some time, is one of the key. Available since about 2012, the detector can capture quick fire images of an individual molecule at dozens of frames per second. These have given cryo-electron a very significant move. The view that the technique would not achieve much better resolution was shared by many researchers about cryo-EM. Perhaps these accounts illustrate the substantial efforts needed to bring cryo-EM to today's level and why these developments have been referred by Werner Kulbrand as the resolution revolution. I am showing you there are many like examples nowadays, but I will show you the structural details of HIV envelope. You will see that started the year of 2003 to 6 to 8 to 2017, the resolution become higher and higher. First one was greater than 4 nanometer at 2003, it comes 3 nanometer at 2006, it comes 2 nanometer at 2008 and 0.36 nanometer 3.6 angstrom at 2017. The rocketing number of cryo-EM publications suggests this to be true. In 2015 alone, the technique has so far been used to map the structure of more than 100 molecules, but in 2018 it even surpassed in number of structural entry in a single year to the number NMR have achieved. So, that is a significant one. And unlike X-ray crystallography, in which crystal lock proteins in a single static pose, researcher can use cryo-EM to calculate the structure of a protein that has been flash frozen in several configurations and so deduce the mechanism of how it works. So, crystallography is definitely giving you a very high resolution structure, but cryo-EM is talking about the biology and now in same resolution. In May, structural biologist John Rubinstein at the University of Toronto and his colleague used around 100,000 cryo images to create a molecular movie of a rotor shape enzyme called V-ATPase. So, this is enzyme V-ATPase and this is the molecular movie you are looking at, developed by them taking 1 lakh micrographs. So, it is not only now a 3D development, it is 3D movement development that is also added here in the technique of cryo-electron microscopy, giving us more understanding. So, V-ATPase pumps proton in and out of the cell vacuoles by burning ATP. What we saw is that everything is flexible. If you look at the protein, you will understand what Rubinstein is saying. Everything is flexible, which is a new mode of vision we are getting by the involvement, by the advancement of the technique of cryo-electron microscopy. It is bending, twisting and deforming. I do not need to show you, you could see the movie presented by them. He thinks that the enzyme's flexibility helps it to efficiently transmit energy released by ATP to the pump. So, now from a high resolution structure, you could also go for determination of understanding of function directly. The solution of ribosome structure by X-ray crystallography won 3 chemists the 2009 Nobel Prize in Chemistry. Venki Ramakrishnan is one of them, Thomas Steed. So, they got Nobel Prize for the structure of ribosome, but in addition to that, the rhizomania has gripped cryo-EM researchers. Why? Because ribosome is a stable complex and is good enough in size to go for cryo-electron microscopy, and it is very important because it is the protein synthesis machinery. So, various teams have quickly determined and published dozens of cryo-electron microscopy structure of ribosome from a multitude of organisms, including the first high resolution model of the human ribosome. As I told, X-ray crystallography has largely fallen by the wayside in the LMB laboratory of Venki Ramakrishnan, who shared the 2009 Nobel. For large molecules, it is safe to predict the cryo-EM will largely supersede crystallography, he says. So, that is where the win of cryo-electron microscopy researchers. One day, they had to listen that they could never compete or supersede X-ray crystallography, they would be a niche and all, and today they are not only providing the resolution revolution at the level of X-ray crystallography, they are also producing structures with much more biological sense, with much better knowledge regarding conformation, regarding activity, regarding its connection to biology, and now it is truly acknowledged by Venki, a crystallographer, that this technique have gone so far and now establish itself as one of the most prominent structure solution technique. With that, I would end up just remembering the Nobel Prize of Chemistry, the work of them. So, as I have already talked about, the Nobel Prize in Chemistry 2017 was awarded to 3 researchers who had spent decades advancing cryo-electron microscopy. You all know the history now. What they did, we talk in details, but to summarize, in 1975, Joachim Frank began work on the algorithms that would analyze fuzzy 2D images and reconstruct them into sharp 3D structures. So, from 2D micrograph, the picture to a model, that is where Joachim Frank stands. In the early 1980s, Jacques Dubochet succeeded in vitrifying water, which allowed the biomolecules to retain their shape in a vacuum. So, Jacques Dubochet, vitrification of water. In 1990, Richard Henderson was the first to use an electron microscope to generate a 3D image of a protein at atomic resolution. So, that is about cryo-electron microscopy, their advancement, and I have done comparison in each level with the other two techniques. I will finish up with the future prospects. A number of improvements are expected in future: high voltage electron source, mathematical correction of lens defects, the aberrations, more higher resolutions, improved scanners, better high pressure freezing, which is one of the place a lot of work needed, and improved CCD or direct electron detectors will remove the need for computer processing in future. So, that is about cryo-electron microscopy. As I have told earlier, this is not a course where I am teaching you cryo-electron microscopy. The idea of this course is to make you knowledgeable about structural biology, is to know you about the basics of this technique, and I have tried to communicate, tried to make you excited, and I am ending up saying, look at this technique. This is a technique for you to look at, because this is a technique which will definitely contribute significantly in the world of structural biology. Thank you very much.